So imagine for a second that you need this really life-saving medical procedure.
Right.
But to actually get it, your doctors have to bring your body to the absolute brink of destruction.
Yeah, it's terrifying.
It really is. I mean, they basically have to carpet bomb your biological systems, like severely damaging your organs, totally wiping out your fertility, and just significantly raising your risk of developing cancer decades later.
All just to save your life in the short term.
Exactly, just to save you. It sounds like something out of, the dark ages of medicine.
Yeah, it really does sound medieval. But sadly, this is the current everyday reality for patients who have to undergo haematopoietic stem cell transplant.
Yeah.
It's just a brutal, albeit, highly effective process.
Which is exactly why we're here today. Welcome to today's deep dive. We are jumping into a massive stack of research, primarily centered on this frankly groundbreaking Nature paper.
Oh, it's a huge paper.
Yeah, it's titled Non-Genotoxic Transplantation and In Vivo Selection Through Epitope Editing. And our mission for this deep dive is to explore this literal sci-fi level molecular trick these researchers have developed.
It really does feel like sci-fi.
It totally does. And it's a trick that can make toxic chemotherapy a thing of the past for gene therapies. So, okay, let's unpack this. Why exactly do we have to practically, destroy the body to save it during a bone marrow transplant?
Well, it fundamentally comes down to biological real estate.
Yeah.
I mean, if you want to give a patient a new healthy immune system or a new blood system, you first have to physically clear out the old diseased bone marrow.
You have to make room.
Right. In scientific terms, we call this clearing the niche. And traditionally, this is done with genotoxic conditioning.
Genotoxic meaning?
It literally breaks apart DNA. We're talking about high-dose chemotherapy and total body irradiation.
So the sledgehammer approach.
Exactly, the sledgehammer. You're essentially just nuking the entire environment just to clear out a few parking spots in the bone marrow, so the new healthy stem cells can actually take hold.
And the collateral damage from that genotoxic sledgehammer is just immense.
Oh, it's devastating. You're looking at multi-organ toxicity and prolonged periods where the patient has literally zero immune system.
Leaving them vulnerable to just about any passing infection.
Right. Plus all the lifelong consequences we mentioned.
So naturally, scientists have been searching for a scalpel instead of a sledgehammer. And they found one, right? Monoclonal antibodies.
Yes, exactly. Monoclonal antibodies, or MABs. These are targeted therapies. So instead of destroying everything in their path, researchers designed these specific antibodies to act like heat-seeking missiles.
Okay, hunting down a specific target.
Precisely. They hunt down a very specific flag, which is a receptor on the surface of stem cells called KIT.
Also known as CD117, right?
Yep, CD117. When the antibody binds to this KIT receptor, it selectively kills those stem cells, theoretically leaving the rest of the body's organs perfectly safe.
Which sounds great, but the paper points out a fatal flaw with this targeted solution. It's a massive timing problem.
Yeah, it's a huge issue of pharmacokinetics. You see, these targeted antibodies belong to a class of proteins that have a very, very long half-life in the human body.
Meaning they just stick around forever.
Right, because of the way the body recycles them, they can stick around for weeks.
Yeah.
So imagine you give the patient the antibody to clear out their diseased bone marrow.
Okay.
It does its job, the marrow is cleared. And now you inject the beautiful, new, healthy donor stem cells to cure them.
But the toxic antibody is still just floating around in their bloodstream.
Exactly. The lingering antibodies recognize the KIT flag on the new donor cells too, and they immediately destroy the very cure you just injected.
Oh, wow.
Yeah, it's known in the field as on-target depletion.
It's like hiring a highly efficient, totally ruthless cleanup crew to empty out an apartment building so new tenants can move in.
I like this analogy, yeah.
But the cleanup crew refuses to leave the building. So the moment your new, healthy tenants walk in the front door, the crew throws them out the window too.
That is a remarkably accurate way to visualize it. And, you might think, well, just wait for the cleanup crew to leave.
Just wait for it to wash out of the system.
Right. But if you wait too long for the antibodies to naturally wash out of the patient's body, the disease host cells that manage to survive the initial purge, they just rapidly divide and steal all the empty apartments back.
So we are stuck in a total catch-22. The chemo is way too toxic, but the safe antibodies linger too long and kill the cure.
Exactly.
Just to quickly recap for everyone listening, chemo is toxic. Antibodies are safer, but they linger and destroy the new donor cells. So if you've ever tried to solve a problem only to find your solution created this weird timing paradox, keep that in mind, because this paper proposes a totally wild workaround.
It really is wild.
Instead of trying to fire the cleanup crew, what if we just disguise the new tenants?
And that right there is the conceptual leap that makes this research so exciting. To understand how they achieved this molecular disguise, we need to look really closely at that KIT receptor.
The CD117 flag.
Right. You can think of KIT as a vital antenna just sticking out of the surface of the stem cell.
And a stem cell fundamentally needs this antenna, right? It's not something we can just snip off.
No, not at all. The antenna receives a specific natural signal called stem cell factor, or SCF.
Okay.
When SCF binds to the top of the antenna, it sends a crucial signal into the cell telling it to survive, grow, and multiply. Now, this antenna isn't just a straight flat line. It's a complex 3D protein structure.
Made of different folded segments, right? The extracellular domains, or ECDs.
Exactly, the ECDs. So the researchers painstakingly mapped exactly where two specific therapeutic antibodies named Fab79D and SR1 physically grab onto this antenna.
They found the exact molecular handholds.
Yes. For the Fab79D antibody, the handhold is on domain 4, and for the SR1 antibody, it's on domain 2. Once they knew the precise physical location where these antibodies grabbed on, they decided to change the physical shape of those handholds using advanced gene editing.
Ah, okay, so this is the epitope editing part of the paper's title.
Exactly.
An epitope is basically just the specific piece of the antigen that an antibody attaches to. So they are designing a molecular invisibility cloak.
That's essentially it. To dodge the Fab79D antibody, they used a tool called adenine base editing. They went into the genetic code and changed a single amino acid in that domain for handhold.
Just one amino acid.
Just one. They converted a histidine to an arginine at position 378. We call this the H378R mutation.
Let me just stop you right there because I want to make sure we really grasp the mechanism. How does changing just one single amino acid out of hundreds make a cell invisible to a heavy-duty drug?
It comes down to protein folding and electrical charge. A protein is, well, it's like a highly complex origami shape.
Right.
Histidine and arginine have different molecular weights and different electrical properties.
Right.
By swapping just that one piece, you change the physical bulge and the magnetic charge of that specific spot on the antenna.
And the antibody is like a puzzle piece designed to fit the original shape perfectly.
Exactly. When you change the shape and the charge, the antibody simply bounces off. It can no longer grip.
But wait, if we mutate the antenna, doesn't that break its ability to receive that vital stem cell factor we talked about? I mean, if the antenna is deformed, the cell should just die anyway.
And that is the absolute genius of this hack. They found a mutation that completely breaks the antibody's grip, but it doesn't break the antenna itself.
Oh, wow.
The KIT receptor still perfectly binds to its natural survival signal, the stem cell factor.
Okay, let me try to visualize this. Imagine the stem cell is a house, and the KIT receptor is the front door.
Okay, I'm following.
The natural survival signal, the stem cell factor, has a master key that fits perfectly into the top lock. But the toxic antibody is a burglar with a skeleton key that fits into the bottom lock. Epitope editing is basically just calling a molecular locksmith to change the bottom lock. The burglar's key no longer fits, so the drug can't get in to destroy the house. But the natural survival key still works perfectly on the top lock to keep the cell alive.
That is a fantastic analogy. The receptor has multiple binding sites, and the researchers just surgically altered one of them. Now, you can give a patient a heavy dose of the targeted antibody. It will hunt down and kill all the unedited, diseased host cells.
But when you inject the edited donor cells,
They're wearing that invisibility cloak. The drug bounces right off them. They peacefully settle into the bone marrow, receive their natural survival signals, and completely take over.
it fundamentally flips the script on bone marrow transplants. So for anyone tracking along, epitope editing alters the specific shape on the KIT receptor so antibodies can't bind, but the receptor still functions perfectly.
Exactly.
So listeners, if you could alter one tiny detail of your digital footprint to become invisible to targeted ads without losing your internet access, would you? It's basically the same concept.
It really is.
But if we think critically about this, if I have, say, sickle cell disease, and you give me invisible stem cells that still have the genetics for sickle cell disease, we haven't actually cured anything.
No, you just have invisible sick cells.
Right. We need a therapeutic payload to go along with the cloak. We have to multiplex.
Exactly. Making the cell invisible is only half the battle. You have to cure the underlying disease at the exact same time.
Yeah.
The researchers needed to perform the epitope, edit the invisibility cloak, and the therapeutic edit the cure simultaneously in the exact same cells.
And the cure they went after is this brilliant piece of biology on its own. They targeted something called the BCL11A erythroid enhancer.
Specifically the plus 58 and plus 55 sites. Yeah.
I'd love to break down how this works, because usually when we think of a genetic disease, we think of a broken gene, and the cure is to fix or replace that broken gene. But that's not what they're doing here at all, is it?
No, it's actually a fascinating double negative mechanism. Let's look at sickle cell disease. The disease is caused by a mutation in the adult haemoglobin gene.
Right.
When you are in the womb, you don't use adult hemoglobin. You produce fetal hemoglobin, which is incredibly efficient at carrying oxygen, and crucially, it doesn't sickle.
But shortly after you are born, your body produces a repressor protein called BCL11A.
Exactly. BCL11A acts like the brakes on fetal hemoglobin. It shuts it down, forcing the body to switch over to the adult hemoglobin, which, in these patients, is broken.
Okay, so the researchers thought, what if we just remove the brakes?
Yes. The BCL11A enhancer, those plus 58 and plus 55 sites, is the genetic switch that tells the body to produce the BCL11A repressor.
So if the therapeutic edit acts like a hammer and smashes that enhancer, the body stops making BCL11A.
Right. The brakes are removed, and the body is forced to revert back to producing the healthy, protective fetal hemoglobin it made before you were born.
You are literally breaking a genetic element that forces the disease to manifest. It effectively cures the disease phenotype by replacing the broken adult hemoglobin with perfectly functional fetal hemoglobin.
It's incredible, right? Breaking a gene to cure a disease.
It's wild. So the researchers applied both of these edits, the invisibility cloak and the broken brakes, simultaneously to human stem cells in a petri dish. And they used some highly advanced CRISPR tools to do this. I saw acronyms like SPIRY-ABA and PE3 in the paper. Can we unpack those mechanisms?
Certainly. These are next-generation gene editors. They use base editing, specifically SPIRY-ABA, for the base substitution we talked about earlier.
The H378R mutation.
Right. Traditional CRISPR acts like molecular scissors. It cuts both strands of the DNA, which can be somewhat toxic to the cell. Base editing is more like a molecular pencil eraser.
Oh, that's a good way to put it.
It uses a deaminase enzyme to chemically convert a single DNA letter like an A into a G without cleanly cutting the double helix.
So it's much gentler on the cell. Much
gentler. And of the other antibody evasion mutation, the one dodging the SR1 antibody, they use prime editing, or PE3.
To make the D121L or S123P mutations.
Exactly. If base editing is a pencil eraser, prime editing is a word processor doing a highly precise search and replace. It uses a guide RNA not just to find the target, but to physically provide the new genetic text.
And then a reverse transcriptase enzyme actually writes the new sequence into the genome.
Yes, it's incredibly sophisticated.
But I mean, if you are deploying two distinct complex molecular machines into a cell at the exact same time, isn't there a massive risk that the cell just dies from the stress? Or that, one edit works and the other totally fails?
That was a major concern going in.
What are the odds a single cell successfully gets both the cloak and the cure?
Well, they proved a profound biological synergy here. They found that these edits naturally co-occur at a remarkably high rate in single cells.
Which changes everything about how we filter the cells. Here's where it gets really interesting.
Exactly. Because they highly co-occur, if a cell survives the antibody treatment in the body, which it can only do if it successfully received the KIT invisibility cloak, it almost certainly carries the BCL11A therapeutic cure as well.
The conditioning drug itself actively filters and selects for the cured cells.
Yes, it links survival directly to success.
Just a quick recap. Researchers multiplexed the edits, combining the invisibility cloak with the BCL11A disease cure. Think about how powerful it is to link survival to success. Where else in life or technology do we use a mechanism where the only way to survive a filter is to also carry the solution? It's brilliant.
It's an elegant solution to a really messy biological problem.
But as we all know, what works beautifully in a sterile petri dish doesn't always translate to a living, breathing body.
No, and that is the ultimate hurdle in translational medicine. To prove this actually works, the researchers moved to in vivo testing using a specialized animal model called NBSGW mice.
Why these specific mice? I mean, what does NBSGW even stand for?
Well, the acronym relates to their specific genetic knockouts, but the important part is that they are profoundly immunocompromised. They are specifically bred to accept human stem cell transplants without their own immune system attacking and rejecting the human cells. This allows scientists to study human blood biology inside a living organism.
Okay, so they took these mice and gave them a mixed transplant. Some of the human stem cells were unedited, representing a patient's natural diseased cells.
Right.
And some were the multiplex edited cells containing both the cloak and the cure. Then they hit the mice with the antibodies, Fab79D or SR1. What happened?
Well, they mapped out different dosing schedules to really understand the pharmacokinetics. They discovered that extended treatment regimens were the key. When they gave the mice the antibody every 5 days or every 10 days, referred to in the paper as Q5D or Q10D regimens, they applied constant selective pressure.
And the result of that pressure?
Massive progressive enrichment of the edited cells. The invisible cells didn't just survive, they completely took over the myeloid and erythroid lineages.
Wow.
Yeah, those are the parts of the blood system responsible for producing red blood cells and driving the immune response. They totally dominated.
Okay, I have to play devil's advocate here because whenever I hear about scientists artificially forcing a tiny handful of mutant cells to rapidly multiply and repopulate an entire biological system,
You get nervous.
I get incredibly nervous. Aren't we risking clonal expansion? Like, what if just one of those edited cells is slightly rogue, and because we gave it this massive survival advantage, it takes over the whole body and essentially causes leukemia?
That is the exact critical question any hematologist would ask, and is why the researchers performed a very elegant safety check using lentiviral barcodes.
Yes, the barcoding. Walk us through the mechanism of how they tracked this.
Sure. Before they transplanted the cells into the mice, they used a lentivirus to insert a unique degenerate DNA sequence into the genome of each individual stem cell.
When you say degenerate sequence, what does that actually mean?
In genetics, it basically means a mathematically randomized string of nucleotides. Think of it like a random barcode generator. It stamps a unique serial number on every single cell. So you have millions of unique serial numbers.
Okay, so we have millions of tag cells. Fast forward weeks later, the edited cells have taken over the blood system. How does the barcode tell us if we caused leukemia?
They extract the blood and sequence those barcodes. If your fear was correct and one rogue clone had outcompeted everything else to cause leukemia, they would see millions of copies of just one single barcode.
Ah, I see.
But to measure what actually happened, they use something called the Shannon Diversity Index.
Wait, I've actually heard of the Shannon Diversity Index used in ecology, right? Like measuring the health of a rainforest?
Exactly. It's the exact same math. In a rainforest, a high Shannon index means you have thousands of different species of trees, insects, and birds coexisting perfectly.
Right.
A low index means you just have a monoculture of, say, one type of pine tree. Applied to stem cells, a high index means the blood is being produced by thousands of different healthy parent cells.
And what did the data actually show?
The Shannon Diversity Index remained remarkably high in the edited cells. The antibody selection didn't skew the clonal diversity at all.
Oh, that's huge.
It really is.
Yeah.
It didn't force one rogue cell to go crazy. It just gently tilted the playing field so that all the diverse, invisible, cured cells could calmly take over the space left by the dying, unedited cells.
That is incredibly reassuring. It proved the expansion is healthy and polyclonal. But speaking of safety, we also have to address off-target effects.
Right. Always a concern with gene editing.
Whenever you edit DNA, there is the risk of the CRISPR tool accidentally binding to the wrong page of the instruction manual and making a cut where it totally shouldn't.
Yeah, and they extensively mapped the entire genome looking for those mistakes. And the results for the prime editing tool, which they used for the KIT mutation against the SR1 antibody, were stunning.
In a good way.
Yes.
Yeah.
It showed absolutely zero detectable off-target genomic substitutions. It was incredibly precise.
0 off-target edits across the whole genome. That is wild.
It is. The adenine-based editing tool, however, did show some minor off-target hits.
Okay, why is that?
Well, this happens because the deaminase enzyme can sometimes be a bit promiscuous and act on sequences that look very similar to the target site. It highlights that while this technology is revolutionary, Continuous optimization of these base editors is necessary before they are perfectly safe for human trials.
Right. Science is always an iterative process, continually refining the tools. But I want to highlight what I think is the absolute mic drop moment of this entire paper.
This sequential transplant.
Yes. Because the new donor cells are totally immune to the antibody drug, the researchers proved you can give the new transplant while the conditioning drug is still heavily circulating in the patient's system.
Exactly. It's a game changer.
Think about what this means for a patient. No more keeping them isolated in sterile rooms for weeks with a zero immune system, just waiting for the toxic chemo or the lingering antibodies to naturally wash out of their blood.
Right. It completely decouples the conditioning phase from the transplantation phase. You can initiate the clearing of the bad cells and plant the good cells practically on the same day.
It fundamentally solves the pharmacokinetic timing paradox we discussed at the very beginning. The drug is actively clearing the niche at the exact same moment the invisible donor cells are moving in. So, to recap this part, in vivo testing proved the antibodies safely enrich the cured cells over time without causing dangerous clonal dominance and allow for flexible transplant timing.
That's a perfect summary.
For everyone listening, what stands out to you about this? Is it the precision of the editing or the clever manipulation of the body's own environment? It's just so much to digest. We've covered a lot of ground today.
We really have.
We looked at how researchers used precise molecular tools to change a single amino acid on a stem cell's receptor, making it invisible to targeted antibodies. We looked at how they multiplexed that with a brilliant double negative edit to cure sickle cell disease, and how in vivo testing proved it maintains healthy clonal diversity without causing leukemia.
It truly is a masterpiece of biological engineering, and it's a testament to how far CRISPR technologies have matured in just a few years.
So, what does this all mean? Zooming out from the amino acids and the lentiviral barcodes, what does this actually mean for the future of medicine? We're looking at a very tangible timeline where curing severe genetic diseases no longer requires bringing a patient to the absolute brink of death.
And if we connect this to the bigger picture, this isn't just about sickle cell disease or beta thalassemia. The authors emphasize that this is a highly modular platform.
It's basically plug and play.
Exactly. You pair the epitope, edit the invisibility cloak with whatever therapeutic edit the specific patient needs. This could be deployed for inborn errors of immunity, where a child is born without a functioning immune system. It could be used for severe bone marrow failure syndromes.
The paper even mentioned targeting leukemias.
Yes. You could theoretically edit a patient's healthy stem cells to be invisible to a targeted leukemia drug. That would allow oncologists to aggressively and continuously treat the cancer with the antibody without constantly wiping out the patient's healthy blood supply.
It fundamentally rewrites the math on risk versus reward for genetic therapies. It takes therapies that were previously considered an absolute last resort because of their massive toxicity and potentially moves them up to a standard of care. That sledgehammer of genotoxic chemotherapy might finally be getting replaced, not just by a scalpel, but by a cloak of invisibility.
A molecular disguise that turns a hostile, toxic environment into a welcoming home for the cure.
I absolutely love that framing. And as we wrap up, I want to leave you listening with a final thought to mull over. If we can use this advanced epitope editing to make engineered stem cells perfectly invisible to targeted immune therapies, how far could we push this boundary?
That's the million-dollar question.
Right. Could we eventually use this exact same molecular logic to protect transplanted solid organs from rejection? Imagine if we could edit a donor kidney or a donor heart so that the host's immune system simply doesn't recognize it as foreign. It could completely eliminate the need for a lifetime of brutal immunosuppressant drugs.
It's entirely possible. The molecular handholds are out there in the human body, waiting to be redesigned.
They really are. So to everyone listening, keep questioning and keep exploring.